It's about 1:30, so we'll go ahead and get started. The purpose of this webinar is we have one on Thursday with the Abbott salespeople, and next Thursday, and they will be discussing how to sell the test, the CT&G test. But going into that, we'll be over basically the science behind the test, so that the webinar can be really sales-oriented, and we don't have to spend time going over the science aspects of it, because when you go into doctors' offices, you want to speak the language and explain it to them. So we'll go ahead and start it. Malcolm isn't with me here; it's just me, Walter McAndrew, and so here we go. We'll begin with the classification of organisms. So you have basically two main prokaryotes and eukaryotes. Prokaryotes are bacteria, and eukaryotes are animals, plants, fungi, protists, all that sort of stuff. I'll let you read over later the differences between them, but that's the first step in division. Then you have viruses. Viruses, it's sort of widely debated whether they're actually living or not, because they basically depend on a host cell to have life. They have their proteins and DNA and everything, but they don't have any machinery to replicate it. So it's sort of a which came first, the chicken or the egg sort of thing. If the virus was first and then used the host, or if it came from the host, that's a whole another topic that we can talk about later. So that is microbiology, and that's specifically pertaining to CNG, which are bacteria. So we have microscopic organisms, the genetic, biochemical, and pathogenic properties, as well as taxonomy, or how you classify and name them. Biology is composed of 4 disciplines: biology, mycology, parasitology, and bacteriology. So we're here at Castle at the moment. We're doing virology and bacteriology testing. We'll be going into mycology for sure in the future, and possibly parasitology as well. But at the time, it's just virology and bacteriology. So each of these, you have commercial, clinical, and then research sectors. So what you now is I'm trying to keep it mostly relevant to the clinical, which treatment of disease. Your research is going to be studying the bacteria themselves, not necessarily for the purpose of curing disease, but just learning more about them. And the commercial is sort of how can we use the bacteria or their byproducts for us in our consumption of goods and that sort of thing. This is just a quick little history to get an idea of how this all started. You have Robert Hooke, and he was the first person to really use a microscope to look at smaller things. He was looking at cork slices, and he's famous for the term cell to describe living organism, microscopic organisms. You have Anton von Leeuwenhoek, one of the first people to really revolutionize the microscope. He made his own, and it's interesting because he actually made it to look at cloth fibers, not organisms, but then realized the potential. And you have Louis Pasteur, who most people have heard about, pasteurization of milk. And he did a test that proved that the reason food spoils is because of bacteria. And he major contributions to germ theory of disease, which basically has given rise to the modern treatment of disease as we know it. And it said that infectious diseases were caused by microbes, and not, back in the 1600s when they thought it could be anything from religious to superstitious and all that sort of stuff. So let's look at bacteriology, or bacterial physiology. So you basically have the cell structure, which the main way of sort of differentiating and classifying bacteria. This includes cell membrane, cytoplasmic membrane, cell walls, all that sort of thing. Then you have cellular metabolism, which is basically what the cells consume to live, so how they produce energy, inputs and outputs. And this, along with cellular structure, this is very important because it sort of determines the function of the bacterial cell in its environment. And then replication. All bacteria basically replicate by making an exact copy of themselves, so splitting into two. The rate at which they do that and what happens when they do that can determine their pathogenicity and basically how bad of an infection you could have with a certain bacteria. So earlier about how cell structure was the major way of classifying bacteria. So there's essentially two types, and it's based on a staining procedure that's, the first thing you would learn in a microbiology class, and it's called Gram staining. And basically, you use a series of dyes and fixatives to prepare a microscope slide. And gram-positive bacteria, if you look at the diagram down here, you saw a wall, this pink. That on gram-positive bacteria has layers of peptidoglycan, which essentially act as layers of shielding from the outside. So positive peptidoglycan will absorb a purple dye, and so when you look at them under a microscope, they will be purple. This is gram-negative, and they lack that peptidoglycan on their cell wall. A lot of times they'll have an outer membrane or an envelope, but do not have as thick of a structure. And so the dye with them will actually, the purple dye will not attach, and a red one will. So look at them under a microscope, they will be red. And so it's kind of interesting that, you think just colors, but it actually tells you a lot about the physiology of the bug you're looking at in a microscope, which type it is, because whether or not they have that peptidoglycan layer will determine their either resistance or susceptibility to a lot of different types of antibiotics. Chlamydia. Chlamydia trachomatis is, tells you the taxonomy right there, gram-negative, obligate intracellular parasite, so that means it needs a host cell to survive. It has a biphasic life cycle, and it's infectious during one stage. This is why you can see a lot of latent infections or asymptomatic infections, where people actually have the bacteria, but they won't be exhibiting normal clinical symptoms that you would expect. And so one interesting thing about chlamydia is they form things called elementary bodies, which is like a mold spore. This is just that diagram. Shows you sort of how they do it, and typically they'll do this under times of stress. They'll make these little mold spores, and so that's why these can be particularly difficult to treat and take a few rounds of antibiotics sometimes because they can be pretty persistent. Neisseria gonorrhea, there's the taxonomy right there. It is a gram-negative as well, and it's facultative intracellular, which means that it doesn't necessarily need a host cell to survive, but it does better and thrives more in host cells. Neisseria gonorrhea will survive a little longer in its normal cellular form outside of a host cell than gonorrhea. The interesting thing here is, and you might hear this in the field as well, a lot of people refer to them as gonococci or gonococcus because they're always found in pairs, and that's actually a wider term, but it's often used interchangeably with Neisseria gonorrhea. Now that's up to molecular diagnostics. So in molecular diagnostics, what we're doing is we're testing for specific nucleotide sequences that are associated with the disease. So that could be a sequence from a virus or a bacteria, or it could be a sequence in a mutated human gene. And it involves testing DNA or RNA from bacteria, viruses, and humans. And another interesting thing, or a thing to note, and you'll hear this a lot, especially talking with people in the industry, qualitative and quantitative assays. So a really important thing to remember, qualitative, you're basically testing to see if, is it or not, is bacteria or virus there, is a mutation there or not, yes or no, And then quantitative, looking at the levels of either virus, bacteria, or mutation. Now, typically you're going to see quantitative a lot more in infectious disease than you will with human testing like oncogenetics, but still often we'll see quantitative testing for human mutations like JAK2, allelic burden, which is basically just a percentage of the mutation. So, molecular diagnostics, there's essentially three major branches. You have clinical infectious disease, genomics, and blood donor screening. Infectious disease, virology, bacteriology, and we talked about earlier, mycology, parasitology would also fall into those. And genomics, which is around human gene testing. So like here, like Varsha is doing oncogenetics, pharmacogenetic testing, genetic diseases and disorders. And then blood donor screening, that's blood typing, and you're basically testing the blood cells of an individual for what antigens are on the outside, and that sort of determines the blood cell type. So molecular diagnostics process or assay has three fundamental steps to it, and it starts with a sample input and it produces a result. So whether testing for a human mutation or you're testing for an infectious agent, you're going to follow that process to get from sample to your answer. So first step would be the extraction and purification of your nucleic acids, so your DNA or your RNA, from interest, whether it be human or a microorganism. Next, the amplification of the nucleic acid. Basically what happens here is you're making multiple copies of the target gene that you're looking at. Be the detection of that target gene after you've amplified it and labeled it with some sort of dye. And that amplification term is also what PCR is, and that's done by real-time PCR, and then there's a lot of other PCR procedures and technologies, but what specific system for CT and G uses is real-time PCR, so that's really the only thing I'm going to delve into today. So we'll start with extraction. So this isn't quite exactly how it works on the M2000, but this is a pretty good representation. You start a sample, you add a lysis buffer. So lysis is the rupturing of cell walls. So you rupture your bacterial cell walls. You do not add proteinase K. Proteinase K basically just cleans up the extraction and eats up free proteins. So your DNA has no proteins in it. DNA can be surrounded by proteins and have proteins on it, but there are no proteins in your actual DNA, so it's okay to eat those up. Then add magnetic beads to the solution. And here is you create an environment in the solution where beads have a certain charge that attract the DNA, and the DNA will actually stick to the surface of the beads. So what you do is apply a magnet to the tube. It's going to pull the beads out of the solution. You take that solution off, so now all you have left are your beads with your DNA or RNA. They wash it with a solution that basically keeps the DNA or RNA on those beads. So you're purifying it and removing any other junk that might be in there, so at the end all you have left is DNA RNA on the beads. And next you will basically elute the DNA and RNA off the beads. So we'll put in a chemical that interrupts that interaction between the beads and the DNA and RNA, and then you're left with just pure nucleotide. So that purified DNA or RNA, the next step would be going into a PCR reaction. So this is just your basic PCR reaction, not necessarily a type of PCR reaction that happens on the M2000, which is the machine we use for CT and G. So a polymerase chain reaction, basically, if you look at this diagram, one copy of DNA is split into two. And in this diagram, you'll see the blue and the red. So you can track the blue, the original copy, all the way down. Now, basically what this does is it allows us to take that one, if you look at the blue, very small amount of DNA and amplify it to a level that's actually detectable. because you just looked at this solution with an analyzer and you didn't do PCR, you wouldn't be enough DNA to detect. So we amplify it exponentially, and they have a level that is easily detected. So the specific type of PCR technology or the method we're using is called TACMAN. So in PCR, you have three basic steps of the reaction. You have saturation, annealing, and extension. So TACMAN uses a quencher and a fluorophore. So what happens is you have this primer. So if you look at the diagram here, the green and the red, the primer is by itself. Nothing happens. It's just sitting there. It's not fluorescing. So if you look at it on an analyzer, you wouldn't see anything. So the first thing you do, you heat it up. That causes it to go from double-stranded into single-stranded. So now that primer can come down and sit on it. Hold on, let me see if I can. Yeah, okay. So you have the primer down, sit on the DNA, and so these nucleotides will only match up with the specific sequence they're programmed for. So in our case, we're specifically looking for genes in CT and G, so it will only sit down and match up with those genes. So the next step is called annealing, and what you do is you lower the temperature and allow that primer to go ahead and match up and connect to your target sequence. So this is very important. This is where you'll hear a term called specificity, and specificity is basically how accurate is matching between base pairs. So then next step is going to be extension. Next step, you have Taq polymerase. So polymerase is a family of enzymes that you see in DNA, RNA replication. And what polymerase does is it goes along and it this nucleotide, and then it grabs one and puts matching one on it, and it goes down the line making the DNA. So the TACMAN, the way it works is as it's now replicating down and making a new strand of DNA, it releases the fluorophore. It's no longer next to the quencher dye that was keeping it from pressing. Now it sends off a wavelength that is detectable. And with cycle, as it occurs, you'll be able to see a greater and greater amount of fluorescence released from the reaction. So this is how we actually detect whether or not an organism is there by the level of fluorescence. So basically looking at, okay, DNA to say that it is definitely there. So the system we're using, it's called the Abbott M2000 real-time system. It is a pretty much automated system. It is one of the best on the market. It is consists of basically two pieces. The M2000 SP. So what the SP does is it's automated, so it's going to extract your nucleic acids from the sample. It's going to purify them, and then it's going to put them on a PCR plate, which is basically where your reaction is going to take place. Then it's also going to put master mix on that same plate, which is what contains your primers and your polymerase, which are essentially the gas and the engine of the reaction. So the M2000 SP does that. The other part of the real-time system is the M2000 RT instrument. This is your analyzer, and it's also what's called a thermocycler. So all PCR takes place on a thermocycler, which is essentially a heat block. And if we back, I talked about these steps of PCR. So this is a very high temperature. This is a low temperature, usually about 6 degrees Celsius, and it's sort of a mid-range temperature, usually about 70 degrees Celsius. Cycling of temperature is going to take place on your thermocycler. So that's going to happen here. And then it also has a camera here that's going to read the fluorescence put out by the reaction in real time. So I didn't mention this earlier, but one of the reasons we call it real-time PCR is that if you look down here, with each cycle, we read the fluorescence. For each cycle, the camera essentially takes a picture and say, hey, at this cycle, here's the fluorescence level. And so you can track it, and it'll actually make a graph with a curve. I should have put one of those on here. But that's how we detect the fluorescence. Thing that's unique to our platform is the multi-collect collection kit, CT&G, and like when I was at Northside, the Roche system that used CT&G, and there was like 3 different collection kits, and we used to have to reject samples. You had a male collection kit, a female collection kit, and a human collection kit. So what they did is they took all of this and put it into one kit. So it's really great because basically it allows the collectors to not have to worry about using the right one. When it's here, we don't have to reject it and send it back and say, sorry, we know you took this, but now you need to recollect because it's not the right thing. So it's a really great aspect of the system and a great selling point, but I won't go into that. We'll let the Abbott people talk about that next Thursday. So, why are we talking about this? Well, you have sexually transmitted diseases, which are a major problem, and as the population grows, so will the problem of STDs. In 2008, the CDC said that there were 19 million new cases of STDs within the U.S., not even globally. 1 million new cases of chlamydia, and 700,000 new cases of gonorrhea in 2008. And that's not even counting people who have not been diagnosed. Their estimate is about, there's half of that value that are probably going undiagnosed. Other things about chlamydia and gonorrhea is they're a notifiable disease. So the CDC and the NNDS require that they have a list, and you can find this on the CDC website, of which diseases are notifiable. And if a patient tests positive, you have to call state officials, local and state health officials, and notify them that basically demographics of the patient, that they were positive. It doesn't mean that they're going to come down with an infectious team and isolate the person. Basically, what they're trying to do is monitor the spread of these diseases and their prevalence within certain populations to try to prevent them and look at, okay, this area has had an outbreak or an increased number of CT&G infections. Let's do public education there or something like that. So it's really more of an institutional thing. CT is the most common notifiable disease in the United States, and then G is the second most. So, these are very common infections and very prevalent in certain populations. Some facts about STDs. So, CT, why do you test for it? Why is it such a problem? So, one of the main things with these 2 infections, both can be asymptomatic, which is a big problem when you're trying to control the spread of a disease, because if people don't think they're sick, they're not going to go to the doctor. In these, 30% of women with untreated CT infections develop pelvic inflammatory disease, which can lead to infertility and other health disorders. And especially in women, CT women has a very, very high rate of asymptomatic infection because the symptoms are mild or nonspecific and so often misdiagnosed or not really considered a problem at all. Infections, second most common, and there also can be asymptomatic, lead to pelvic inflammatory disease, infertility, ectopic pregnancy, and chronic pelvic pain. With both of these microorganisms can increase your risk of contracting other venereal diseases as opportunistic pathogens. So, not necessarily an STD, but they essentially weaken your immune system and make it easier for other infections to get in and take hold. So that's for today. Sorry if I went too fast. Feel free to know if you have any questions. And then also I put some here to CDC sources. And then Go Molecular is actually a site set up by Abbott. Talks a lot about molecular diagnostic testing and all is into it, basic background, how it works, that sort of thing. So thanks, guys. And like I said, next Thursday, we will have the people from Abbott come, and they will also be giving a webinar. They will talk specifically about how to sell CNG and sort of selling points of our system in particular. So hope you join me for that one as well. Thanks.